Hi everyone, good evening. So welcome to this series, which is a score booster series that is organized by PrepLadder for NEET PG 2025 aspirants. And this is a session on PCR, polymerase chain reaction and detection of known mutation sites. Yeah. So, so have you seen questions in this topic? Right, there are so many questions which are being asked in this topic. For example, they ask you the enzyme that is used in polymerase chain reaction is. Your answer should be Thermus aquaticus DNA polymerase. Or they ask you questions like, what is the technique that you will use to detect a known mutation site, right? And what is real-time PCR? Of course, they don't ask you straight away questions like what is real-time PCR. They ask you about SARS-CoV-2 diagnosis. So for you to be able to answer such questions, I'm going to help you understand the concept behind this topic. So what is the overview for this session? In this session, I will be first telling you what are the types of cloning, what is PCR, what is RFLP, restriction fragment length polymorphism. I will also be telling you what is denaturant gradient gel electrophoresis, single strand conformation polymorphism. What are the techniques that can be used to detect known mutation sites? And apart from this, I will also tell you the differences between reverse transcriptase PCR and real-time PCR. So this is an overview for this session. Let's start with what is cloning and what are the types of cloning. Cloning is nothing but production of identical copies, right? So here we are trying to produce identical copies of DNA. Why or when will you want to produce identical copies of DNA? It has got many applications. First, it has got genetic engineering application. For example, if you're trying to produce insulin on a large scale, any protein for that matter, if it needs to be produced on a large scale, the first raw material that you would want is insulin gene as multiple copies. Only then you will get multiple mRNAs of insulin, then you will get multiple copies of insulin protein. So cloning DNA has got many applications. One is genetic engineering application. The other one is forensic application, wherein from the crime site, maximally what you can expect is a hair follicle. And from the hair follicle, before you can, not just hair follicle, right? It can even be a blood spot. And from hair follicle or a blood spot, maximally you will expect 1 or 2 copies of DNA. And before you can perform any DNA fingerprinting technique, what you're supposed to do first is you get multiple copies of the DNA from the blood sample or from the hair follicle. Then you can perform any DNA fingerprinting technique. So this is about where we are going to use cloning DNA. One is genetic engineering, the other one is forensic application. But as a process, if you ask me what is cloning DNA, it is nothing but producing multiple copies of DNA or replicating DNA multiple times. And for replicating DNA multiple times, we have few requirements, right? So what are the requirements? The first requirement is you need an enzyme which replicates. And that enzyme is called as DNA polymerase. And apart from DNA polymerase, all DNA polymerases need primers. So the next requirement is primers. We need all 4 deoxynucleotide triphosphates because DNA polymerases cannot synthesize a new DNA out of nowhere, right? All that these DNA polymerases can do is they look at the template strand. Depending upon the sequence that is present in the template strand, they just recruit complementary nucleotides and they link complementary nucleotides. So you have to provide them with those complementary nucleotides. That is why they need all 4 deoxynucleotide triphosphates. They need magnesium or manganese to act as a catalyst. They need a buffer to maintain the pH. So if you are asked what are the requirements of replication, your answer should be DNA polymerase. All DNA polymerases need primers. You need all 4 deoxynucleotide triphosphates, magnesium or manganese to act as a catalyst, and a buffer to maintain the pH. Now, depending upon what is going to provide you with all these requirements of replication or all these requirements of cloning, cloning is of 2 types. One is if a whole living cell like E. coli provides you with all the requirements of cloning, then that is called the cell-based cloning, which is nothing but recombinant DNA technology. So what do we do in recombinant DNA technology? We take the gene of interest, we attach the gene of interest with a vehicle which is called as a vector. Then we introduce that recombinant vector into E. coli. Then E. coli provides with all the requirements of cloning. Cloning is happening within a cell. That is called as cell-based cloning or recombinant DNA technology. On the other hand, if you don't want to meddle with a whole living cell, what you can do is you can take a small test tube, you add all the requirements of cloning, and if cloning happens in vitro, that is called as in vitro cloning, which is nothing but enzyme-based cloning, and that is polymerase chain reaction. So what are the two types of cloning? If cloning is happening within a whole living cell, that's called as cell-based cloning or recombinant DNA technology. If cloning is happening in a small test tube or polymerase chain reaction tube, right, PCR tube, then that is called as in vitro cloning or enzyme-based cloning, which is polymerase chain reaction. So the purpose of this session is for you to understand all about PCR. Okay, so if I ask you to define PCR, will you agree with me if I say that PCR is an amplification process? You know that, right? PCR is an amplification process. But for this term amplification, you need to add two adjectives. One is, it is in vitro amplification, because here amplification is happening in a test tube. So it is in vitro amplification. Second adjective is based on the fact that it is not a random amplification. You amplify a specific fragment. So if you're asked to define PCR, your answer should be, it is in vitro amplification of a desired fragment of DNA. And if you're asking me, how do we specify what we want? We specify what we want by constructing specific primers. So how do you choose these primers? We choose these primers in such a way that these primers are complementary only to the flanking sequences of the desired fragment, right? Suppose this is the desired fragment, then what I do is I choose primers in such a way that these primers are complementary only to the flanking or the terminal sequences of the desired fragment. So that is all about definition of PCR. So what is the definition of PCR? Polymerase chain reaction is an in vitro amplification of a desired fragment of DNA. And we manage to synthesize only a desired fragment by constructing specific primers in such a way that these primers are complementary only to the flanking sequences of the desired fragment. Okay. Now about steps of PCR. And I want you to follow the steps of PCR, imagining that we are involved in molecular diagnosis of sickle cell anemia. Give me a second. So why am I specific about sickle cell anemia? It's because sickle cell anemia is caused by a known mutation site. So what do I mean by a known mutation site? All over the world, sickle cell anemia is caused only by mutation of the 6th codon, right? It's caused by mutation of the 6th codon of beta globin gene. Do you ever say that sickle cell anemia in Asia is caused by a mutation of 6th codon, whereas in Africa it is caused by mutation of the 8th codon, whereas somewhere else it's caused by mutation of an intron? No, we don't have such variations. Sickle cell anemia all over the world is caused by only a mutation of the 6th codon of the beta globin gene. Right, 6th codon of the beta globin gene. So this is called as a known mutation site. So if you are involved in detection of sickle cell anemia, then I want only a fragment of beta globin gene. So how will I get exactly what I want? I start with the entire DNA. Suppose these two lines represent the entire DNA. And these red lines, yeah, do you see red fragments in the center? This red fragment represents fragment of beta globin gene, which includes the 6th codon. Now to get exactly what I want, I'm going to subject it to PCR, which is a replication process. And you know, for any replication, the first step is unwinding, right? For any replication, the first step is unwinding. So what is the first step of PCR? The first step is you unwind the double-stranded DNA to form two single strands. Can you see that here? I've unwound a double-stranded DNA to form two single strands. Unwinding is otherwise called as denaturation. So tell me, what is the first step of PCR always? Is that? Yeah. So can you please listen to what is being discussed here? Because PCR or polymerase chain reaction is a high yield topic. Okay. And of course, it's given in many textbooks and in many guides. But practical application of these techniques are not explained in any of these books. And what is being questioned these days is it's only application aspect. Okay. So in this session, I will tell you what is PCR. I will also tell you how is it applied. Okay, so what is the first step of every cycle of PCR? The first step of every cycle of PCR is unwinding or denaturation. And this unwinding or denaturation is carried out at a very high temperature of 94 or 95 degrees. Okay, after unwinding, in the next few steps, your DNA polymerase is going to elongate new strands. And for your DNA polymerase to elongate new strands, you know that you need primers. So in the next step, I'm going to add two primers, and these two primers will be complementary to the flanking sequences of the desired fragment. What do I mean by flanking sequences? Terminal sequences of the desired fragment. So in the next step, I'm adding primers, and these primers are annealing to the flanking sequences. So this step is called as annealing step. So what should be the temperature of annealing step? The temperature of annealing step has to be optimal. It is 5 degrees less than the melting temperature of your primers. Please memorize it. Annealing temperature is variable because it is 5 degrees less than the melting temperature of your primers. For example, if melting temperature of one primer is 64 degree and if melting temperature of the other primer is 62 degree, can you tell me what is the average between 64 and 62? It is 63 degree. So average melting temperature being 63 degree. What is 5 degrees less than 63? It is 58. So for this example, annealing temperature will be 58 degree, but it is variable. It varies based on the very good. Yes, you're right. So it varies based on the melting temperature of the primer that is being used. Okay, so if you're asking me why should it be 5 degrees less than the melting temperature of your primers, it is because if it is more than the melting temperature of your primers, then primers will not anneal and primers will denature, right? It should not be more than the melting temperature of your primers. But at the same time, it should not be too less than the melting temperature of your primers, because in that case, your primers do not anneal to specific sequences. Your primers start annealing somewhere else, and you will get something called as non-specific amplification. So to avoid non-specific amplification, annealing temperature should be neither too high nor too low. It has to be 5 degrees less than the melting temperature of your primers. The second fact about annealing step is, for annealing step, you need to add two primers. These two primers should be complementary to 3' flanking sequences of the desired fragment. Please be specific. They should be complementary to 3' flanking sequences of the desired fragment. For example, if AB represents the fragment that you need, right? AB represents the fragment that you need. And suppose the top strand is in 3', 5' direction. For AB, A represents the 3' flanking sequence. B represents the 5' flanking sequence. How have I chosen primers? I've chosen primer in such a way that it is complementary only to the 3' flanking sequence, only to A. Why? It's because DNA polymerases can synthesize a new strand only in 5', 3' direction. Even if you don't know this till now, please memorize it. Any polymerase It can be DNA polymerase, it can be RNA polymerase. All your polymerases can synthesize a new strand only in 5' 3' direction. So only when your primer is complementary to 3' flanking sequence here, the primer hybridizes in which direction? 5' 3' direction, and your DNA polymerase elongates what you need. Okay. Hypothetically speaking, suppose by mistake, I choose primers. Okay. So by mistake, I've chosen primers in such a way that they are complementary to 5' flanking sequences. In that case, you know that your primers go and hybridize here. And the direction of synthesis is 5' 3'. So you will be getting something which you don't need. To avoid that, you should choose 2 primers. Both the primers should be complementary only to the 3' flanking sequences of the desired fragment. That is about annealing step. In the next step, elongation. Your DNA polymerase is going to elongate new strands. So the next step is called as elongation. And for this elongation, you need a thermostable DNA polymerase. A DNA polymerase which is capable of withstanding cyclical rise of temperature to 94 or 95 degree. So whatever step I'm telling you now represents the various steps of every cycle of PCR. So in one cycle of PCR, there is denaturation where you raise the temperature to 94 or 95. Again in the next cycle, 94 or 95. So the DNA polymerase that you use for polymerase chain reaction has to be able to withstand a cyclical rise of temperature to 94 or 95 degree. That is why we have extracted a thermostable DNA polymerase from an organism which lives in hot springs. And that organism is Thermus aquaticus. What is the name of the organism? The name of the organism is Thermus aquaticus. And the DNA polymerase is called as Taq DNA polymerase. And the optimum temperature that is required for Taq DNA polymerase to elongate a new strand is 72 degree. That is why elongation step is always carried out at 72 degree. Are you clear about this? So tell me, what are the 3 steps of every cycle of PCR? Denaturation, annealing, elongation. Denaturation is carried out at 94 or 95 degree. Annealing is carried out at what temperature? 5 degrees less than the melting temperature of your primers. Elongation is carried out at 72 degree. The DNA polymerase that you need for polymerase chain reaction is Thermus aquaticus DNA polymerase or Taq DNA polymerase. This way, if you amplify a particular fragment through n number of cycles, at the end you can expect 2 to the power of n products. Suppose you set the PCR reaction for 30 cycles. At the end of 30 cycles, you get 2 to the power of 30 products. 35 cycles, you get 2 to the power of 35 products. Such a sensitive technique is PCR. Okay. And if you ask what is the machine that is used for polymerase chain reaction, the machine is called as thermocycler. So can you all repeat this? What is the machine that is used for polymerase chain reaction? It is thermocycler. Okay. And during the second wave of COVID pandemic, we realized that it is not practically possible or feasible to install thermocyclers everywhere. And even when we were ready to invest money to install all these thermocyclers in the periphery, getting people with technical expertise to handle thermocycler was a huge challenge. That is when we realized that there is a need for isothermal amplification technique. First, tell me, why is this machine called as thermocycler? All that the machine does is it cycles between temperatures. First, it increases the temperature to 94 or 95 degree. Then it reduces the temperature to 5 degrees less than the melting temperature of primers. Then it increases it to 72 degree. Again, it rises to 95. So all that the machine does is it cycles between temperatures, and that's very difficult for having a machine which is able to cycle between temperatures. So we are in need of an isothermal amplification technique. And that isothermal amplification technique is called as lamp assay. So what do you mean by LAMP assay? LAMP assay is loop mediated amplification assay. Okay, LAMP assay is loop mediated amplification assay, for which you cannot use Thermus aquaticus DNA polymerase. We need a special DNA polymerase which is extracted from an organism called as Bacillus stearothermophilus. Yeah, Bacillus stearothermophilus or Bacillus smithii. Have you heard of Bacillus stearothermophilus? Does that ring a bell? You must have learned this as an organism which is used as a bioindicator, right? For autoclave, the bioindicator that is used is Bacillus stearothermophilus, and DNA polymerase that is extracted from this organism is called as BST DNA polymerase. From Bacillus smithii, it is BSM DNA polymerase. And the speciality of these DNA polymerases is that they have very high strand invasion ability. So you don't have to raise the temperature to 94 or 95 degree, just increase the temperature to 60 degree. So what happens at 60 degree? At 60 degree, there will be mild denaturation happening at various places in your DNA. That by itself is enough for these two DNA polymerases to invade the strand and to elongate the strand. So every step in isothermal amplification technique or in LAMP assay will be carried out at a constant temperature of 60 degree, for which you don't need a thermocycler, just an incubator would do, which can maintain a constant temperature of 60 degree. Do you understand this? So that is about what is a LAMP assay. Okay. So you don't need any technical expertise to handle this LAMP assay. Anybody who can make a tea will be able to use LAMP assays. Got it? So that's all about what is conventional PCR and what is LAMP assay, which is isothermal amplification technique, which uses BST DNA polymerase or BSM DNA polymerase, which allows the entire reaction to get over at 60 degree. Okay. Now about what are the steps to be followed for detecting a known mutation site? What do I mean by a known mutation site? I gave you an example. It is sickle cell anemia, which is always caused by mutation of the 6th codon of beta globin gene. So whenever the mutation site is known, okay, you have to follow a sequence of steps. Step number 1 is it's a molecular diagnostic technique. For any molecular diagnostic technique, step number 1 will be sample collection. In this case, I'm collecting a blood sample. Okay. Step number 2 is DNA extraction. Okay. Once I've extracted DNA, whenever I say I've extracted DNA from a blood sample, I mean to say that I've extracted all 46 chromosomes of the individual and I have all 25,000 genes of the individual. Do you understand this? Whenever you say you've extracted DNA, you mean to say that you've extracted all 46 chromosomes with all genes. But for detection of a known mutation site, for example, sickle cell anemia, do you want to struggle with this entire genomic material? No. I want only one gene. Which gene do I want? I want only beta globin gene. And even beta globin gene, I don't want the entire gene. I'm going to be happy taking a fragment of beta globin gene, which includes the 6th codon. So how will I get exactly what I want? I will get exactly what I want by subjecting the DNA that I have extracted to PCR. So after DNA extraction, step number 3 is subjecting it to polymerase chain reaction, wherein I choose specific primers in such a way that these primers are complementary only to the flanking sequences of a fragment of beta globin gene, which includes a 6th codon, right? Following PCR, I get a PCR product. What is this PCR product? It's a fragment of beta globin gene which includes the 6th codon. Still, what I don't know is, even after PCR, I don't know whether the 6th codon is normal or is it mutated. So the take-home message for all of you here is, conventional PCR will not directly give you the diagnosis. Conventional PCR will help you to only get a fragment that you want. And then to find out whether it is normal or mutated, you have to subject it to few other techniques. One of the few other techniques. A straightforward next step would be sequencing. Right after PCR, if I want to know whether the sixth codon is normal or abnormal, what I can do is I can subject it to sequencing. Okay. And the limitation of using a sequencer is sequencers are not widely available and sequencing is not a cost-effective option, right? And that is why we are in a hunt, right? We are in a hunt for some other simpler technique or affordable solution. So what are the other affordable solutions? Second one is RFLP or restriction fragment length polymorphism. Third one is single-stranded conformation polymorphism. What is the third one? Single-stranded conformation polymorphism. The fourth one is denaturing gradient gel electrophoresis. The last one is denaturing gradient gel electrophoresis. So after PCR, what are the four choices you have to find out whether it is normal or mutated? Straightforward answer is sequencing. But we have RFLP, single-stranded conformation polymorphism, denaturing gradient gel electrophoresis. Let me start with what is RFLP. Okay, we are going to learn about all these techniques now. So RFLP, as you all know, is restriction fragment, right? What is it? It is restriction fragment length polymorphism. And this technique uses an enzyme. And that enzyme is called as restriction enzyme. So for RFLP, we always use an enzyme which is called as restriction enzyme. What are restriction enzymes? All restriction enzymes are endonucleases which cut at specific sites. Do you understand this? All restriction enzymes are restriction endonucleases which cut at specific sites. So how are we going to choose a restriction enzyme for RFLP? You cannot randomly choose any restriction enzyme. There are thousands and thousands of restriction enzymes available in the market, but you have to choose a restriction enzyme in such a way that the enzyme cuts the mutation site. Yeah, you have to choose a restriction enzyme in such a way that the enzyme cuts the mutation site only when it is normal. If it is mutated, the enzyme should not cut it. That is how you choose a restriction enzyme. And then, depending upon whether it is cut or uncut, you will be able to give the diagnosis. Okay, let me give you a specific and simple example that I've been giving you. Molecular diagnosis of sickle cell anemia, right? So following PCR, I have this fragment of beta globin gene, which includes a sixth codon, the length of which is 1.4 kilobase pair. To make it simple, I have chosen primers in such a way that I get a fragment of beta globin gene, length of which is 1.4 kilobase pair. Okay. And I know that the sixth codon is cut by a restriction enzyme by name MST2. So we have an enzyme by name MST2 which cuts the sixth codon only when it is normal. So if it's normal, 1.4 kilobase per fragment is cut to give rise to two fragments, one of length 0.2, the other one of length 1.2 kilobase pair. If it's mutated, it will not be cut. It will stay as 1.4 kilobase per fragment. This information I already know. So how will I use this information for diagnosis? I subjected the DNA that was extracted to PCR. Following PCR, this is the PCR product that I have, the length of which is 1.4 kilobase pair. Into this PCR tube, I add the restriction enzyme. What is the name of the restriction enzyme? MST2. And I incubate this tube. Okay. And during this incubation period, I expect the MST2 to cut it if it's normal. It will not be able to cut it if it's mutated. Right. After the incubation period, to find out whether it is cut or uncut, I run these fragments through electrophoresis. So what I'm drawing here is a gel. And in this gel, we are running electrophoresis. This gel has got 3 wells. Do you understand? This gel has got 3 wells and I have 3 samples. Patient A, patient B, patient C. I'm adding patient A's restriction digest into well 1, person B's restriction digest into well 2, person C's restriction digest into well 3. And I run it through electrophoresis. I know that longer fragments cannot move, shorter fragments move faster. Do you understand this? Longer fragments cannot move and shorter fragments move faster. And person A's restriction digest moves only till this. I know if a fragment moves till this, the length is 1.4 kilobase pair. I already know if the fragment moves only till this, the length is 1.4 kilobase pair, which means it is not cut. What does it mean if it's not cut? It means it is mutated. In this case, I know that this person has got mutation. Okay. Person B's restriction digest moves and stops here. I know that if a fragment stops here, the length is 1.2 kilobase pair, which means it is cut. If it's cut, it is normal. Okay. Look at person C's restriction digest. Person C's restriction digest gives me two bands. One corresponding to 1.4, the other corresponding to 1.2. So why am I getting both 1.4 and 1.2? Let me remind you of the fact that all our genes, we have two copies. Yes or no? For every gene in your body, you have two copies. So when I say one is cut, the other one is uncut, it means one allele is normal. This is a normal allele and this is a mutated allele. So person C is heterozygous for the mutation. Do you understand this? Person C is heterozygous for the mutation. Person C, the genotype is AS, meaning person C is a carrier. Okay. What is the genotype of person A? Both are uncut, which means the person A's genotype is SS or the person is sickle cell diseased. What is the genotype of person B? Both are cut, which means both are normal. So the genotype of person B is AA. So person A is sickle cell diseased, person B is normal, person C is heterozygous for the mutation, person C is a carrier. Do you all understand this? If you have understood this, can I see a thumbs up, please? This is about how RFLP can be used for disease diagnosis. Do you all understand this? So how do you use RFLP for disease diagnosis, all of you? Hi, Nikita. Others, have you all understood? So tell me the steps involved in RFLP. For RFLP, step number 1 is sample collection. Step number 2 is DNA extraction. Step number 3 is you subject the extracted DNA to polymerase chain reaction. And then step number 4 is you're subjecting the PCR product to digestion by a restriction enzyme. And depending upon whether it is cut or uncut, you will know the diagnosis. Okay, that's all about RFLP. Now, apart from RFLP, I said there is a third choice. And what is the third choice? The third choice is single-stranded conformation polymorphism. So what is this used for? Single-stranded conformation polymorphism is used following PCR to find out whether a person has got the normal allele or a mutated allele. To find out whether a person has got normal wild type allele or the mutant allele. Okay, so what do we do here? It's very simple. The same set of steps. What will be step number 1? Sample collection. Step number 2 will be DNA extraction. Step number 3 is again PCR. So this PCR has given rise to this allele, right? This allele. And suppose this represents the normal or wild type allele and this represents the mutant allele that is present in the person. This is where the mutation has happened. Okay, so what I do is I subject these two to denaturation, right? What will happen when I denature it? All these will become single strands. The moment they become single strands, all single strands have an intra-chain base pairing tendency. Anything which is single stranded will bind, right? They will fold to form hairpin loops. Depending upon their sequence, they'll fold to form hairpin loops. So that gives them a specific conformation. Do you understand this? That gives them a specific conformation, and the specific conformation that you get from single strand will differ depending upon the sequence. When the conformation differs, when you run it through electrophoresis, the way they migrate will also be different. Okay, so what do we do after PCR and single-stranded conformation polymorphism? You denature the fragments. You're not going to add a restriction enzyme. You denature the fragments. Once you denature the fragments, they all take a characteristic shape. The shape they get will differ depending upon the sequence. Based on the shape they have, electrophoretic migration will be different. So look at this. So this image tells you what happens in single-stranded conformation polymorphism. This is the wild type allele or the normal allele. This is the mutant allele. This is where there has been a mutation. When I denature, they are all single strands. Once they become single strands, because of intra-chain base pairing tendency, they fold to get a characteristic shape. Do you see that the characteristic shape is always different because the sequences are different? Okay, so in a person, when you run it through electrophoresis, if the migration mimics this, I know this is normal. Okay, if it migrates like this, I know it is mutated. If you see both kinds of migration, it means the person is heterozygous for the mutation. So depending upon how it migrates after denaturation, I will be able to make a diagnosis of whether a person has got normal allele or a mutated allele. Is that clear? So that's about single-stranded conformation polymorphism. The fourth choice is denaturant gradient gel electrophoresis. What is this used for? This is also used to find out specific sequence changes following PCR. Okay, so the basic difference between single-stranded conformation polymorphism and denaturant gradient gel electrophoresis is, in single-stranded conformation polymorphism, in the PCR tube itself, you mediate denaturation and then you run it through electrophoresis. Whereas in denaturant gradient gel electrophoresis, following PCR, the PCR product will be run through electrophoresis. Do you understand this? The PCR product will be run through electrophoresis and the gel will have increasing gradient of denaturant. The gel of electrophoresis will have increasing gradient of denaturant. Okay, and depending upon the sequence change, the point at which denaturation happens will be different. For example, if the normal allele has AT, but if the mutant allele has GC, okay, AT has double bonds, GC has triple bonds. So which will undergo denaturation faster? The normal allele will undergo denaturation faster. As soon as there is denaturation, the migration stops. Right? As soon as there is denaturation, the migration stops. So if I see that person A's restriction digest stops here, quickly it stops. It means it has undergone denaturation quickly in the gel, which means it has AT. If there is AT, it is normal. If I see that it has moved till this, it means it has GC, which means it's a mutant allele. Do you get it? So what do we do in denaturant gradient gel electrophoresis? In denaturant gradient gel electrophoresis, we allow the PCR product to run through electrophoresis, the gel of which has increasing gradient of denaturant. Increasing gradient of denaturant. And depending upon the sequence, the point at which denaturation happens differs. If there is AT sequences, denaturation happens faster and it stops there. If there are GC sequences, denaturation happens later and the migration is more. So that's about denaturant gradient gel electrophoresis. Okay. So whenever they ask you, what are the steps to be followed to make a diagnosis of a known mutation site, you should never get confused. Step number 1 is sample collection. Step number 2 is DNA extraction. Step number 3 is conventional PCR. PCR does not directly give you the diagnosis. To find out whether it is normal or is it mutated, we have 4 choices. What are the 4 choices? Sequencing, restriction fragment length polymorphism, single-stranded conformation polymorphism, and denaturing gradient gel electrophoresis. Okay, now let's learn about what is RT-PCR, because SARS-CoV-2 diagnosis is done by this technique, right? So RT-PCR for all practical purposes is reverse transcriptase PCR. And do not get confused with real-time PCR, because real-time PCR is otherwise called as quantitative PCR. Because it is otherwise called as quantitative PCR, the acronym that is given for real-time PCR is Q-PCR. Reverse transcriptase PCR, the acronym is RT-PCR. For real-time PCR, the acronym is Q-PCR. So what are the differences between these two techniques? We use reverse transcriptase PCR when the nucleic acid of interest to you is an RNA. Do you understand this? We use reverse transcriptase PCR when the nucleic acid of interest to us is an RNA. Example is SARS-CoV-2 diagnosis, because SARS-CoV-2 is an RNA virus. If you are interested in an RNA, you cannot directly amplify RNA. You cannot directly quantify RNA. If you're wondering what is amplification, what is quantification, amplification is producing multiple copies. Quantification is you quantifying the number of nucleic acid. So RNAs cannot be directly amplified. RNAs cannot be directly quantified. You can only amplify and quantify DNA. But if the nucleic acid of interest to you is an RNA, you should first convert this RNA to DNA. That is done by reverse transcriptase. I hope you all know that DNA to RNA is transcription. Yes or no? DNA to RNA is transcription. RNA to DNA is reverse transcription. So whenever the nucleic acid of interest to you is an RNA, you should first convert it into a DNA. That is done by reverse transcriptase enzyme, and the technique is called as reverse transcriptase PCR. Okay. Now, what is the use of real-time PCR or quantitative PCR? This is used for quantifying nucleic acid. And if you're asking me when or why will I want to quantify nucleic acid? To understand pathogen load, right? If you want to understand what is the viral load or what is the bacterial load or what is the fungal load, instead of being dependent on culture and sensitivity based assays, which are always time dependent, right? It takes its own sweet time for you to do culture and sensitivity to quantify it, right? Instead, what you can do is you can quantify nucleic acids and correlate it with viral DNA or bacterial DNA or fungal DNA. That is the purpose of quantifying nucleic acid. That is done by real-time PCR or qPCR. Now, let's try to understand about reverse transcriptase PCR and real-time PCR by understanding the steps that need to be followed for molecular diagnosis of SARS-CoV-2 infection. Okay, let's try doing that. But what will be step number 1 for molecular diagnosis of SARS-CoV-2 infection? Step number 1 is, yes, swab sample collection, right? So we collect both oropharyngeal and nasopharyngeal swab sample. Then the swab sample is kept in a medium. That medium is called as viral transport medium. This viral transport medium is Hank's balanced salt solution. Most of the times it is Hank's balanced salt solution, and it is made up of calcium and magnesium with heat inactivated bovine serum albumin. What is it made up of? Calcium, magnesium, and heat inactivated bovine serum albumin. As you can see here, this medium is loaded with nutrients. So what will happen when you add a swab sample into this medium? Because this medium is rich in nutrient, any pathogen or any, not even a pathogen, any organism that is there in the oropharynx or nasopharynx will start growing. So any bacteria or any fungus that is present in the swab sample will start growing, and that will not allow the virus to survive. So what are you supposed to do? Kill the bacteria, kill the fungus. And that is why we spike the medium with gentamicin and amphotericin B. The medium is spiked with gentamicin and amphotericin B. And after you keep it in the viral transport medium, it should be transported in cold chain, right? If you're asking me why should it be, I am very sure you must have all seen that the swab sample after collection is transported at a temperature of 2 to 8 degrees, right? In a thermocol box, it is being transported. That's called as cold chain maintenance. And the reason for cold chain maintenance is we are handling an RNA virus. So by mistake, on the way, if the RNA undergoes lysis, or RNAs will come out, right? If the virus undergoes lysis, RNAs come out. RNAs are very labile and that will cause false negative reports. So you have to maintain cold chain so that the virus does not undergo lysis until it reaches the lab. In the lab, once a sample is received, we extract RNA. In the lab, once a sample is received, we first extract RNA. When I say I'm extracting RNA, what do you think I would have extracted? I would have extracted all human RNAs from the swab sample because it's a human swab sample. I would extract all human RNAs. Any other viruses present in the swab sample, their RNAs will also be extracted. If at all there is SARS-CoV-2 infection, that RNA will also be extracted. So please remember, RNA extraction is a non-specific step wherein we extract all RNAs present in the swab sample. After RNA extraction, we subject the RNA to reverse transcriptase PCR because, as I told you, RNAs cannot be quantified or amplified directly. So I subject it to reverse transcriptase PCR. I convert the RNA to DNA. Then I subject the DNA to real-time PCR. And in both these places, I specify what I want. Both in reverse transcriptase PCR and in real-time PCR, I use specific primers. In real-time PCR, I use specific primers and probes. They are complementary only to sequences of SARS-CoV-2 nucleic acid. And that is why we call this molecular diagnostics as the most specific and the most sensitive technique. Do you all understand this? So this is about steps involved in molecular diagnosis of SARS-CoV-2 infection. So let me repeat these steps for your benefit. What is step number 1? Swab sample collection. Place it in a viral transport medium. Transport it in cold chain. It is received in the laboratory wherein we first extract RNA. We extract all RNAs. Then we subject the RNA to reverse transcriptase PCR. Then we use those DNAs by real-time PCR to quantify the nucleic acid. Okay. Now more about real-time PCR. So real-time PCR or quantitative PCR is so called because it quantifies nucleic acid real time. As and when amplification happens, quantification also happens simultaneously. That is why it is called as real-time PCR. For your better understanding of real-time PCR, let me tell you this. Real-time PCR is a very late addition. We got it probably 15 to 20 years back. Until then, we were using only conventional PCR even for quantification. Okay, but what we do in conventional PCR for quantification is we would allow the PCR to get over. After everything is over, we quantify. Okay, so look at the graph that is shown to you here. Whenever you're shown a graph, always check what is along the x-axis and what is along the y-axis. That by itself will tell you all about the graph. For example, in this graph, tell me what is along x-axis. Along x-axis, it is number of cycles, and along y-axis, it is DNA concentration. Okay, and the initial phase is called as an exponential phase or a lag phase. And the reason behind why I'm calling it as an exponential phase or a lag phase is initially, in spite of there being an increase in the number of cycles, number of products do not increase that much. Do you see that? In spite of there being an increase in the number of cycle, number of products do not increase that much. That is why it is called as an initial lag phase or exponential phase. If you're asking me why is there a lag to start with, it is because initially the number of template DNA available is less. Initially, there will be 1 or 2 copies of SARS-CoV-2 DNA. From 1 or 2 copies, you can expect only 2 or 4. From 2 or 4, you can expect only 4 or 8. From 4 or 8, you can expect only 8 or 16. So initially, the number of template DNA available is less. That is why the rate of increase in the number of product initially is slow. That's why it's called as a lag phase. Then there is a rapid linear phase, wherein as and when there is an increase in the number of cycle, number of products quickly increase. Because in this phase, the number of template DNA available is optimal. The number of primers, the number of deoxynucleotide triphosphates, magnesium, manganese, everything that is necessary is optimally available. And that is why there is a rapid rise. And then why do you see a plateau finally? It finally reaches a plateau because at this point, all raw materials have been used up. So that even when there is an increase in the number of cycle, number of products refuses to increase. So what are the 3 phases of PCR? Any PCR goes through these 3 phases. The initial phase is a lag phase or exponential phase. And then there is a rapid linear phase. And finally, there is a plateau phase. And in conventional PCR, we used to allow the PCR to get over. In the plateau phase, we would quantify the number of nucleic acid present. From here, we would extrapolate to how much would have been there initially to give rise to so many products at the end. Right? We would just extrapolate to how much would have been there initially to give rise to so many products at the end. But this graph tells you that irrespective of the number of copies of template DNA present in the initial sample, all these samples finally give rise to the same number of products. So from here, if you're trying to extrapolate, right? From here, if you're trying to extrapolate, you would either underestimate or overestimate the number of copies of template DNA available. So this is not the right phase for quantification. The plateau phase is not the right phase for quantification. Which is the right phase for quantification? The initial lag phase or exponential phase is the right phase for quantification. And real-time PCR is so called because it quantifies real time in the initial lag phase or exponential phase. Do you understand this? Why do you call real-time PCR as real-time PCR? Because it quantifies real time. As and when amplification happens, quantification is also done in the initial lag phase or exponential phase. And for this purpose, we use something called as Stackman probe. For real-time PCR, we use something called as Stackman probe. So all of you look at this because even a layman knows real-time PCR is used for diagnosing infections, right? But as medical professionals, we should know more than what they know, right? So we should know what is Stackman probe. We should know what is CT, right? Actually, even a layman would say higher CT means it's better. Lower CT is severe, right? But we need to know what is Stackman probe, what is CT. And that's what I'm going to tell you in the next 3 minutes, right? So watch this carefully for the next 3 minutes for you to understand entirely about real-time PCR. So in real-time PCR, we use Stackman probe. This Stackman probe has got 3 parts, okay? The first part is an oligonucleotide sequence. Yes, it's a live session. So the first part is an oligonucleotide sequence. And this oligonucleotide sequence is not a random sequence, okay? So this is an oligonucleotide sequence which is complementary to mid sequences of nucleic acid of interest to you. Which is the nucleic acid of interest to you now? It is SARS-CoV-2 nucleic acid. So SARS-CoV-2 nucleic acid will have multiple genes. You take one gene. In that gene, mid sequences, complementary to mid sequences of the gene, you synthesize oligonucleotide sequences. That is part number 1. Okay. And this oligonucleotide sequence is attached to part number 2, which is the reporter dye. Why am I calling it as reporter dye? Because this dye fluorescence is what the machine measures. RT-PCR machine, no. Real-time PCR machine measures the fluorescence of only the reporter dye. That's why it's called as reporter, because it reports fluorescence. The third part is a quencher dye. What is a quencher dye? As long as the quencher dye is an attachment with the reporter dye through this oligonucleotide sequence, the quencher dye quenches the fluorescence of the reporter dye. Do you understand this? As long as quencher dye is an attachment with the reporter dye through an oligonucleotide sequence, the quencher dye quenches the fluorescence of the reporter dye. Which means when you have this TACMAN probe, the fully integral TACMAN probe does not show any fluorescence. Why do you think it does not show any fluorescence? Because the quencher dye quenches the fluorescence of the reporter dye. So all of you tell me, what are the 3 parts of TACMAN probe? The 3 parts of TACMAN probe are: number 1, oligonucleotide sequences, which are complementary to mid sequences of nucleic acid of interest to you. And that is attached to a reporter dye, the fluorescence of which is being measured by the instrument. And the quencher dye, which quenches the fluorescence of the reporter dye. This TACMAN probe is added to all other requirements of PCR. So can you tell me what are the requirements of PCR? That is where we started the session, right? So what are the requirements of PCR? Any PCR will need Taq polymerase enzyme. It needs primers. What are primers? Primers are complementary to the flanking sequences or the terminal sequences of the desired fragment. So primers are complementary to the flanking sequences. Probes are complementary to the mid sequences. So you take a tube, you add all the requirements of PCR. What are the requirements of PCR? Taq DNA polymerase, primers, deoxynucleotide triphosphates, magnesium or manganese to act as a catalyst, buffer. Along with that, you also add this TACMAN probe. Okay. And if the sample has got SARS-CoV-2 nucleic acid, this is how everything will go unhybridized. So this is SARS-CoV-2 nucleic acid, and in the SARS-CoV-2 nucleic acid, to terminal sequences primers will bind, to mid sequences probe will bind. Okay, and Taq DNA polymerase will start elongating the primer. That is its function, right? What is the function of Taq DNA polymerase? It is to elongate the primer. So when it elongates the primer, on its way, it comes across TACMAN probe. And your Taq polymerase is not happy to meet a TACMAN probe on its way. It considers its TACMAN probe as an obstacle to its elongation. So Taq DNA polymerase uses its exonuclease activity to break this oligonucleotide. Okay, it does not like an obstacle on its way. So Taq DNA polymerase uses its exonuclease activity to remove these oligonucleotides. Once it breaks this oligonucleotide, do you see that the reporter dye is detached from the quencher dye? Yeah, when the Taq DNA polymerase breaks down the oligonucleotide sequence, reporter dye is detached from the quencher dye. Now, reporter dye is no more in attachment with the quencher dye, and reporter dye starts fluorescing. And this fluorescence intensity is what the machine measures. So, please watch this carefully. The fluorescence intensity that is measured is directly proportional to number of reporter dyes that are detached. Okay, number of reporter dyes detached is directly proportional to Taq polymerase activity. This Taq polymerase activity is directly proportional to number of template DNA that is present. Yeah, it is directly proportional to number of template DNA present. That is why fluorescence intensity is directly proportional to template DNA that is available. Do you understand this? So this is all about real-time PCR. So what are the facts that you need to know about real-time PCR? Please remember, real-time PCR or quantitative PCR is used to quantify nucleic acid. And it quantifies nucleic acid in which phase? In the initial lag phase or exponential phase. For that, it uses Tachman probe. Tachman probe has an oligonucleotide that is attached to a reporter dye and a quencher dye. Tachman probe does not fluoresce because reporter dye's fluorescence is quenched by the quencher dye. And it is added to all other requirements of PCR. So when Tach polymerase elongates, it starts breaking the oligonucleotide that is present in the Tachman probe. So reporter dye is detached from quencher dye, then it starts fluorescing. So fluorescence intensity is directly proportional to the number of reporter dye detached. That is directly proportional to Tach polymerase activity. That is directly proportional to number of template DNA that is available. Okay. Now, in real-time PCR, the machine draws a graph. The same graph which I showed you previously. What is along x-axis? I told you. Along x-axis, it is number of cycles. Along y-axis, it is DNA concentration or the fluorescence intensity. And can you see that it goes through a lag phase, a rapid linear phase, and then a plateau phase? Okay. And the machine has something called as threshold fluorescence. Yeah, the machine has something called as threshold fluorescence or the background or the basal level of fluorescence. Okay, that's called as threshold fluorescence. Now, what is CT? Listen to this carefully. CT in real-time PCR is the minimum number of cycle that is necessary to cross the threshold intensity. Yeah, let me repeat this. What is CT? CT or cycle threshold. So what is CT or cycle threshold? It's a minimum number of cycle that is necessary to cross the threshold intensity. Suppose I say CT of a sample. I am saying that CT of a sample is 35. What does it mean? It means it took 35 cycles. If I say CT is 35, I mean to say it has taken 35 cycles for the sample to cross threshold intensity, which means the number of template DNA available is less. Okay. Suppose I say CT of a sample is only 8 cycles. It means even at the 8th cycle, it has crossed the threshold intensity. Even at the 8th cycle, it has crossed the threshold intensity, which means the number of template DNA available is more. Okay, so CT and template DNA concentration are inversely proportional. Higher CT meaning lower viral load. Do you understand this? Higher CT meaning lower viral load. CT and viral load are inversely proportional. So that's all about real-time PCR. So are you clear about polymerase chain reaction now? Yeah. So these are the facts that you need to know about PCR or polymerase chain reaction. Now let's discuss few one-liners and MCQs here. The first one is the instrument used for PCR is. What is your answer? Can I see answers in the chat box, please? What is the instrument that is used for PCR? It is thermocycler. The enzyme that is used in conventional PCR is. What is the name of the enzyme that is used in conventional PCR? Thermus aquaticus DNA polymerase or Tach DNA polymerase. The enzyme used in LAMP assay is, right? LAMP PCR uses Bacillus stearothermophilus. Very good. Yes, it is Bacillus stearothermophilus DNA polymerase or BST DNA polymerase or Bacillus smithii DNA polymerase or BSM DNA polymerase. Very good. Excellent. Real-time PCR quantifies in which phase? Which phase? Can you all answer? Real-time PCR quantifies it in the initial exponential phase or in the lag phase. MCQs. SARS-CoV-2 diagnosis. Excellent. I'm happy that you're answering. SARS-CoV-2 diagnosis is done by all except. Yes. It's an INI-CET November 2022 question. SARS-CoV-2 can be diagnosed by reverse transcriptase PCR, real-time PCR, and immunoassays. I'm sure you must have all seen card tests, right? Card tests for diagnosing SARS-CoV-2. wherein again you're supposed to collect a swab sample. It's a self collection of swab, and this swab has to be kept in a processing solution. Okay, it has to be kept for a particular time. And then you take out the swab, you will be given with a filler. So you take this fluid using a filler and you add the fluid into one end of the slide. Once you add this fluid, the fluid will start moving. If there is SARS-CoV-2 in the sample, SARS-CoV-2 antigen will react with SARS-CoV-2 antibodies which are there on the card. So there will be antigen antibody reaction. This antigen antibody reaction will give rise to a pink color. So if you see a pink color, it is positive. It is antigen antibody reaction based assay. So it's an immunoassay. So for SARS-CoV-2, we have molecular diagnostic assays and immunoassays. And if you're asking me application wise, what is the difference? As I told you, molecular diagnostic technique will be the most sensitive and the most specific. Yes, it's going to be the most specific because I specify that I want SARS-CoV-2 twice. Once in the form of reverse transcriptase PCR, the other one in the form of real-time PCR. For both these, I use specific primers, and for real-time PCR, I use specific probes. So it's the most specific. Why am I saying molecular diagnostic is also the most sensitive? Because in molecular diagnostics, as I told you, you're amplifying and simultaneously you're quantifying. Do you understand this? You're amplifying the signal and then you're quantifying. So it will be the most sensitive and the most specific. Whereas if it's immunoassay, yes, it will be specific because it is also based on antigen antibody reaction, specific antibody. So it will be specific, but the sensitivity is questionable because here you're not amplifying the nucleic acid, you're not amplifying the antigen. One antigen reacts with one antibody. So immunoassays can be specific, but they are not very sensitive, right? So how do you interpret a card test? When you do a card test for your patient, if it's positive, it is positive. And if the card test says negative, it can still be positive. Do you understand this? If the card test is negative, it can still be positive. So if the clinical suspicion is very high, what will you do? You will ask for molecular diagnostic test even if the card test is negative. So the major advantage of card test is that it's a home-based test. It does not involve technical expertise for sample collection. It does not involve technical expertise to analyze. It's a home-based test, quicker testing, but the sensitivity is questionable. That is about SARS-CoV-2 diagnosis. Okay. Hi, Ansari. How are you? The temperature of denaturation step in PCR is, denaturation is always carried out at 94 or 95 degree. Annealing temperature, I said, is variable. Did I tell you? It is 5 degrees less than the melting temperature of your primers. Right? Annealing temperature in PCR is variable. It is 5 degrees less than the melting temperature of your primers. All the following are requirements of PCR except. Yes, PCR needs DNA polymerase. Right? PCR needs deoxynucleotide triphosphates. It needs magnesium. But what is the right answer? It does not need restriction enzyme. Restriction enzyme is necessary for RFLP, restriction fragment length polymorphism, or for recombinant DNA technology. It is not necessary for PCR. A child with complaints of poor growth, intellectual disability, learning problems was brought to the hospital by his mother. The doctor, after inquiring about family history, sent the patient for molecular diagnosis. Which of the following may not be used? Please understand, may not be used to detect sequence changes in DNA following PCR. So let me wait for you to answer. When you conduct complete LRR for biochemistry. That will be done soon, Crystal. So what will not be used? We will use single-stranded conformation polymorphism. we do use denaturing gradient gel electrophoresis. Dideoxynucleotide triphosphate method is nothing but sequencing. Did I tell you sequencing is a straightforward answer, but it's not available always, and it has got cost, it is very less cost effective, right? So sequencing is done by Sanger's method or by next generation sequencers. Sanger's method is dideoxynucleotide triphosphate method. So following PCR, if you just run it through electrophoresis, that cannot be used. Do you understand that? Yeah, following PCR, if you just run electrophoresis, it will not help you. Following PCR, denaturing gradient gel electrophoresis, yes, it can tell you. Single-stranded conformation polymorphism can tell you. RFLP can tell you. For all these, electrophoresis just a part. But just by running it through electrophoresis, you cannot make a diagnosis. So answer is D. A 21-year-old man wants molecular diagnosis of sickle cell anemia as 3 of his maternal cousins are diagnosed with this condition. The intern knows few steps involved in molecular diagnostics in a jumbled way. Help him choose appropriate steps and arrange in the right sequence. So can you all tell me what is the right sequence? What is step number 1 always? I said for any molecular diagnostic test, step number 1 is sample collection. That should be followed by what? DNA has to be extracted. Then the extracted DNA should be subjected to what? Conventional PCR. This conventional PCR should be followed by RFLP. Right? Should be followed by RFLP. So it is 2764. Sample collection followed by DNA extraction, followed by PCR, followed by restriction fragment length polymorphism. Okay. Image-based question. Look at this. 3 individuals A, B, and C of a family of sickle cell anemia want to get their molecular diagnosis done. With the provided RFLP data, choose the genotype of persons A, B, and C. Okay. So person A, they've given you, this is the restriction site that is lost in sickle cell anemia. If this restriction site is lost, what will be the length of the fragment? The length of the fragment will be 1.4 if it is mutated, right? If it's mutated, the length will be 1.4 kilobase pair. If it is normal, the length will be 1.2 kilobase pair. Now look at person A. The length is 1.2 kilobase pair, which means it is cut, which means it is normal. Person A is normal. Look at person C. What is the length? 1.4 kilobase pair. I explained this in detail, right? So watch the entire video for you to clearly understand the application of PCR and for you to understand what are the steps that need to be followed for molecular diagnosis of a known mutation site. Okay. So this length is 1.4, which means it is uncut. If it is uncut, it is deceased. Person B, one is cut, the other one is uncut, right? One is cut, the other one is uncut. So this is heterozygous. So person A is normal, person B is a carrier, person C is deceased. Normal, carrier, and disease. Answer is choice D. So in this session, we learned concepts related to PCR, right? I told you about types of polymerase chain reaction. We learned about steps to be followed in molecular diagnosis of known mutation site. I told you what is reverse transcriptase PCR, what is real-time PCR. I told you what is Tachman probe. What are the tests that can be done to detect SARS-CoV-2 infection? Okay, so thank you all and see you sometime later.